Now we are going to move on to Dr. Pierre Bellec's talk. Before that, I'll just like to do a brief introduction. I'm not going to take much time. I am Mohit Kumar Jaiswal, and I'm a bioinformatics master student from University of Pune. Now, before getting Dr. Bellec on board, I'd just like to do a brief introduction. Dr. Pierre Bellec is the principal investigator at the CMXP lab, which works at the interface of AI and neuroscience, and is a laboratory for brain simulation and exploration. He's a director of Functional Neuroimaging Unit, Research Center of the Montreal Geriatric Institute, and also an associate professor at the Psychology Department at University of Montreal. He's also co-leading the Discovering New Biomarkers team at the Canadian Consortium on Neurodegeneration in Aging, together with Dr. Roger Dixon from University of Alberta. Finally, Dr. Bellec is the scientific director of the Kotora project on neuronal modeling, NeuroMod, which uses human neuroimaging data to help train large artificial neural networks on a variety of cognitive tasks. And I think it's on a similar flavor that is his talk today, which is titled as the Kotora NeuroMod project, augmenting learning in artificial networks using human behavior and brain functional activity. So Dr. Bellec, it's up to you. Thank you so much. Enjoy, everyone.
Okay, thanks so much for inviting me this great event, NeuroNovember. So today I'm going to give you an overview of the Kotora NeuroMod project. It's a fairly young project. It's 2 years old, and we're now producing our first data release, which I'm going to describe to you. The NeuroMod project is not solely a data producing enterprise. We also have a number of scientific objectives in terms of modeling the human brain. So I'm also going to show you a series of preliminary results we've got on this first data release. But yeah, it's fairly early in the project, and you'll see we're just beginning. But I think this will help you understand why we collect the data we collect and how we collect it. So I should say that the overall objective of this project is brain augmented learning. So deep learning technique, artificial intelligence systems, they're now able to do tasks that until recently we thought were impossible for computers to do. And so that opened new avenues to study intelligence in artificial media. But the way deep learning systems solve tasks, they're often very different from the way humans solve them, both from a behavioral perspective. So what a human finds difficult may be very different from what a computer finds difficult. And also in terms of their architecture. The brain is a very distributed system that's able to shift from many different cognitive contexts and types of tasks very easily, which is something that current deep learning models have a very hard time doing. So the main objective of the project is to try to use those deep learning techniques to imitate human behavior and human brain activity. And we're planning on doing that on fairly complex and multi-domain tasks. And the primary type of task we're going to be looking at are video games. So, video games, I've got this little schematic on the left. It's really like a dynamical system where the brain is constantly exposed to new stimuli and have to constantly decide on action. And so there's a real integration from perception to action, decision-making in the middle. So it's a really great example of this type of integrative process. And this is a type of cognitive processes we're going to try to model primarily. So as an overview of the project, we only have 6 subjects, and we're going to try to characterize those 6 brains very well to make in silico replica of those 6 individuals, at least within the context of particular tasks. There's 3 men, 3 women, and we are planning on acquiring about 500 hours of neuroimaging data on each subject, which would represent roughly 1 order of magnitude more than previous public data sources that we have on human brain activity, at least with the type of neuroimaging data we're acquiring. So I see in the chat that yeah, there's a black, something was obstructing my slides, and that was the chat. I removed it. So now I don't see if you're telling me anything. Sorry. So as I said, we're going to be doing video games, but also other naturalistic tasks and many more controlled experiments in the language, memory domains in particular. And so we're going to be acquiring functional MRI data, magnetoencephalography data, as well as biosignals. So let me tell you a little bit more about those individuals, those 6 individuals. You have to be a little bit crazy to enter that study, because we are scanning the participants once a week for at least 5 years. I'm one of the subjects. That's me with my little equipment to get scanned. Typically, we think about participants in research project as they come, we scan, they go. In that case, I think more of them as the equivalent of astronauts, but for the brain. So the name I use is NeuroNov, because they're really extremely well trained into getting scanned, because they are doing it so often. We also have a number of equipment dedicated to the scanning for those individuals. So that include personalized head case that have been made from scans of their face, so that we limit greatly the amount of motion that can happen in the scanner. So we have those head cases both for the MRI and the MEG. We also have those custom earplugs in silicone to increase comfort and the level of sound protection. Although this is still something we're working on improving, even though we are 2 years in the project, we're still iterating on that aspect. We're also piloting currently those personalized shirts that you can see on the picture. So they've got openings for placing electrodes consistently across sessions. There's also I don't know if you can see it, but there's this little mark on the shirt that allows us to place a respiratory belt consistently across sessions as well. So the 2 equipments we have access to are a Siemens Prisma 3T scanner, which is located at the neuroimaging functional unit at my institution. I'm the director there. We also have a state-of-the-art magnetoencephalography scanner, which is located on campus at the University of Montreal. And so those are the two locations where we scan. One particularity of the project is also that we're acquiring a lot of different biosignals. So that includes measure of cardiac rhythm, both electrical signal, electrocardiogram, and also optical signal with plethysmography. We've got a respiratory belt, as well as measures of skin conductance, which are a reflection of emotional states to some degree. We also have a high-resolution eye tracker. This is something that we're adding now to the project. We don't have it for the first data release, but we're going to deploy it for the second one. So you can track microsaccades with that type of device, and as well as doing pupillometry, so measures of pupil dilation, which is also another marker of emotional states. We do all that because emotions are really critical about the way humans interact with their environment, and are also big drivers of performance, notably in video games, where there's quite a bit of variability, even for a given individual. So we want to be able to characterize those type of emotional states. And as we cannot interrupt the subject constantly to ask them what they think, we're trying to collect as much objective physiological measures as we can. So those data are going to be public. Here I've linked docs.surmo.ca that describes extensively what we collect. I'm at a health institution that is very prudent, very cautious when it comes to sharing human data. And I've been struggling for 2 years with my ethics committee to make the data as widely and accessible as possible, and as easily accessible as possible. Those are very expensive data to collect. I would say that collecting the data for each subject is going to cost about $500,000. just in sort of scanning fees. So I believe the primary aim of the project is to expand research in that area and make it easy for people across the world to study those type of processes and those type of models. So my hope is that in January 2021, we're going to have the data bank opened. So it's going to be a small process to get access to the data. Basically, you need to be registered at a known academic institution, write a short text about your project that we will use to check that this is research about the human brain, basically. And you'll need to sign a data transfer agreement. And from there on, you'll get access to our data bank. It's organized using a standardized file structure, which is a brain imaging data structure initiative. It's also using the data lad system, which means that you have precise control of the versions of the data you're accessing. As well, if you wanted to access to subparts of the data, you can very easily only get this subpart. I mean, those data sets are very large. The full data set will probably be more than 50 terabytes. So, if you want to only get a portion of it, it's possible. And this data will be, all the metadata will be exposed publicly through the Canadian Open Neuroscience platform. Here I've put pictures of our project manager, Dr. Julie Boyle, and our data manager, Basil Pinsard, who are really working hard to make this high quality data resource. So now let's get to exactly what's in the data set. NeuroMod is more a collection of data sets than one data source. Those data sets have in common that they're always the same subject doing everything. Each data set in NeuroMod typically is roughly 10 hours of functional data. For this first release, we don't have the biosignals yet, and we don't have MEG yet. It's only fMRI. So we've got movie 10, which is about 10 hours of people watching movies, so naturalistic stimuli. We've got 4 movies that have been selected to cover very different genres. So we have Born Supremacy, which is an action movie. We've got Life, which is an animal documentary. We've got Hidden Figures, which is a great movie about those women coders that worked on sending people to space in the US. And The Wolf of Wall Street, which is a movie about human greed, capitalism, and misogyny. It's extremely upsetting for some of the viewers. So that's that. We also have Friends, 2 seasons of Friends. Each season of Friends is about 10 hours. So for those who don't know, Friends is like popular series from the 90s, which is coming back into fashion. It's on streaming on Netflix at the moment. The reason why we have Friends is that in total, there's about 100 hours of Friends episode, and they always feature the same actors and the same locations for about 100 hours. So it's great to be able to work on brain decoding, try to understand how the brain responds to those particular individuals, those particular characters, and having lots and lots of stimuli with the same characters. And at the same time, those scenes are relatively interesting. Actually, some people really like the Friends series. So, that was just this very rich set of stimuli with the same scenes, same characters, but engaging scenarios. And finally, we have some more like classical stimuli. The HCP Human Connectome project has developed those tasks that are designed to activate particular locations in the brain. So we've run all those tasks and repeated them 15 times to be able to check whether the systems are reliably engaged. And also we've got some more specific objectives. I'm going to get to that in a minute. So in total for that first release, you have about 40 hours of data per subject. So why did we assemble those particular tasks for that first release? Well, for the first release, we have some key objectives, which are really, we want to understand, as I told you, like the main objective of NeuroMod is to try to mimic brain activity and human behavior with artificial networks. So the first thing we needed to check is whether artificial network can learn to represent the transient functional modules in the brain. And can they learn to decode accurately brain states? So here I have this little schematic of Francisco Varela, almost 20 years ago, but it illustrates how brain connectivity is organized at multiple scale, both local, through the Kotora architecture. Within the gray matter, you have those highly specialized brain areas, but you also have interaction from one side to the brain to the other. And that structural backbone lead to the emergence of synchronized functional assemblies. They're transient, but in a given state of cognition, you get those functional assemblies. So we want to know if we can accurately characterize those at the individual level and detect their presence at the individual level. So the direction we're taking in terms of the modeling is using what's called graph convolutional networks. So those are deep learning networks models that have been proposed about 4 years ago. They've become really popular. Kipf and Welling paper in 2016 has sparked a lot of interest in that. And those type of networks are generalized more convolutional network models. But instead of having convolution based on space in images, here you can use an arbitrary graph to propagate information in data and learn representation from data. And it so happens that we have good priors on graph organization of the brain. And we can use neuroimaging to build a very detailed graph representation of the brain. So the idea that Dr. Yu Zheng had, she did her postdoctoral fellowship with me, was to use brain graphs to try and see whether we could decode what subjects were doing while being scanned. And so that's for me a way to check whether we can, train networks to mimic or at least recognize signatures of brain activity. So we applied that. At the time, we didn't have the Neomat data set, so I'm going to present you results on another public resource that is the Human Connectome Project sample. So we had 1,200 subjects with about 1 hour of functional data per subject. And they were performing many different tasks organized along about 7 big cognitive domains. So you have emotion, language, motor, activity, relational, which is more like matching patterns, social, cognition, and working memory task. And within each of these domains, you had different specific experimental conditions. So in total, you have 20 of them. And what Yu did is train some graph convolutional network to try to detect from short window of temporal activity. Typically here, you look at 7 seconds of fMRI recordings, and the network try to predict what the subjects were doing. And here what you have is a confusion matrix. So on the x-axis, you have the predicted state by the network. On the y-axis, you have the real state in which the subjects were. And you have the probability, the matrix represents the probability of actually being in a given state when you're being predicted to be in that state. And as you can see, we have very large values on the diagonal, 0.8 to 0.9, which means that 80 to 90% of the time, when the network predicts that you're moving your hand, you were actually moving your hand. So we are able to do brain decoding with fairly high levels of accuracy on these tasks. There are some tasks that get mistaken for one another. We can see it here for the relational task. There's some subdivision of the relational tasks you have to do, and those sometimes get confused. About 20% of the times they get confused for one another. So even when the network make mistakes, they're generally not random. It actually confuses cognitive states which happen to be quite similar cognitively. So one of the key questions we had for the Neomat project, like how much data do we need to train this kind of network and to train artificial neural network to recognize states. And here you've got a graph that shows the accuracy of the prediction over those 21 cognitive states as a function of the number of subjects being used in the prediction. And what you can see is that as of about 100 subjects, we are starting to make really accurate prediction. We reach a plateau at around 200. So as I said, there's about an hour of data per subject. So you need roughly 200 hours to train those type of network. Now, bear in mind that this is a brain decoder that applies to any individual. So the cross-validation we apply here is that we train the network on a subset of individual, and we test it on data coming from other individual. So the network doesn't have just recognized brain state. It also has to learn human variability, which is quite large. So I believe this is a very sort of challenging task, but we need roughly 200 hours of functional data to solve that task with high level of accuracy, above 80% overall. Actually, if we optimize the brain graph, we can crack 90% of that task. This is like one of 3 papers that has written on that, and we can even do better than this. But that gave us like this idea that to train networks, we're talking about hundreds of hours. Now we are hoping that if we train network on a particular individual, we can cut down this requirement dramatically. And that's why we repeated 15 times the HTTP task per subject in our first NeuroNov release. So we have 15 hours of those tasks per subject in our current release. And we're hoping to show we're going to be able to train, brain decoder at the individual level. But before we did that, we already had access to one individual data set with several repetitions of the task per subject. And we tried to see if we could take this group model that was trained on the large Human Connectome Project sample and fine-tune it to adapt to a particular individual that with data collected on a different scanner. So we use the individual brain charting projects that is done in France. This is a public data set. There's 12 subjects that repeated the HTTP task twice with slightly different imaging paradigm. And what you have here are different kind of network. On the right, it's just taking the HTTP model and just applying it at this data, just like if they were coming from HTTP with the same scanner as HTTP. And it doesn't work necessarily as well. Like this is just for the motor task where you can just like get really, really high accuracy. And you see that you're only at 90% accuracy if you just use the HTTP model from scratch. But if you allow to fine tune some aspects of the model, on the left, you have all the parameters are free up to only tuning some layers of that model. You can see that you can recover up to 97% accuracy on that task. So I found that very promising, because that means that even if we are not able to train model from scratch on an individual, we could possibly use some group model prior and then fine tune it to each individual to get even better. Now, hopefully, like, in the next few months, we're going to know if we can train network from scratch on individual data on those tasks. And I mean, we already done it. You can do it, but recover this kind of very high accuracy. And currently, we're not able to do it. One of the avenues by which we are hoping to actually get to this very high level accuracies by getting very good individual brain graph. So that's another one of our objectives for that first release. And I'm going to explain to you a little bit what I mean by that. So the graph, I've gone very fast on this, but at the core of a graph representation of brain networks is what's called a brain parcellation. So different regions in the brain that act as little units, and then you have a matrix that describes how those units are connected. So very similar to the explanation I was giving you with the Varela schematic and those local assemblies. You can use data-driven techniques in fMRI to define those brain parcellations, but it became apparent a few years ago, in particular with the Midnight Brain Scan project, that those parcellations are highly individual. So that's a figure from Gordon's paper on the Midnight Brain Scan project. that shows that. So they had about 5 hours of resting state data per subject in that study. And here what you have are the results of those data-driven brain parcellation. And you have different subjects here. They had 10 subjects in that study. And what the arrows point to are highlights of patterns that you find in a specific individual reliably, but that you cannot find in a group average parcellation. So at the top you have the group average parcellation, and here you have different individual. And you can see, for example, like the extent of that blue network at that particular location of the brain is going to be quite variable. I mean, most subjects have one, but if you check where it's located exactly, you can have quite some variation, up to 1 centimeter variation. So when you average that across subject, it becomes very blurry and you lose that information, which is potentially critical to learn good graph convolutional network from scratch on an individual. So those type of variation complicate the task of the group model I was describing. Another thing that became apparent, there's a beautiful study by Salehi and colleagues coming from the Todd Constable lab at Yale, where they tried to generate the type of parcellation I just showed you, but in different tasks. And what they've shown is that there's really stark differences in the parcellation from one task to the other. And what people had always assumed when doing graph representation of brain network is that, to some degree, the units would be fairly stable, and what would change would possibly be the sort of connectivity between those units. But what Salehi and colleagues showed is that there are some really marked variation across tasks, and it's probably not a good idea to neglect those variations. And that better brain graph would take into account kind of dynamic reorganization of parcellation from one cognitive state to another. And really, I mean, I find this result fully unsurprising, because that's what Varela was already describing 20 years ago in his review on functional assemblies. But somehow this is something we're rediscovering with fMRI today. So a PhD student of mine, Amal Boukdir, she developed this algorithm called DIPAC, dynamic parcel aggregation, that looks at small windows of activity, generate those type of parcels for each window, and then aggregate the parcels across all the windows before finding somewhat like reproducible spatial patterns across all the windows. So aggregating those parcels across all the windows. And we're applying that to the movie 10 data. We already have a paper on the technique that was published this year on the midnight brain scan. And I'm going to show you how it works when you apply it to the movie 10 data set. So on the left here, you've got some of those parcels, and they're generated from all the movies of movie 10, so the 4 movies. But what I didn't mention when I presented the release is that 2 of those movies we did twice. So Life and Figures, we scanned a second time. And here what you've got are parcels that are generated from that second repetition of those movies. So completely independent data. That's for one subject. And not all parcels are as reproducible, but those are examples of highly reproducible parcels. And as you can see, the amount of spatial overlap between the test and the retest is very, very high. We get spatial correlation that are above 0.9, 0.95. Some of the regions are less stable within those probabilistic parcels than others. Here you can see that the frontal eye fields are not always associated with those parietal regions, but this is something that we detect reliably. So those regions appear with the same level of stability at the test and the retest. And we are also able to get those parcellation in subcortical areas, here illustrated in the cerebellum. So one way of testing whether there is gain in getting those dynamic and individual parcels, it's to compute a homogeneity of the parcels. So the way it works is that you take your original data, which features about 200,000 voxels, and you project it into a reduced space of brain parcels. Here we had 150 parcels. And then you can compare the time series in the original space with a compressed space based on parcel. And you can get an R-square value. If the R-square is 1, that means the compression is perfect. And if the R-square is lower, then that means that you're losing information. So somebody is raising their hands. So I don't know. Don't hesitate to interrupt me if you have question. I'm going to let the moderators deal with this. But so what we see here is that the compression with individual parcel is of much higher quality than the compression with if you use a parcellation coming from a particular subject to compress the data of that particular subject, you get R-square close to 1, above 0.7, sometimes 0.8 for R-square. So those mean correlation of the original data and the reduced data that are above 0.8 to 0.9. But if you're using parcellations that come from other subjects for those data, so those are the other graph, you see that the values are much lower with R-square values that are in the range of 0.3 to 0.5. So you're losing a lot of the details of the activity of the brain. So basically, those results indicate that using dynamic and individual parcels lead to much better representation of brain activity at the individual level, and thus may help us build much better graph convolutional networks, which is what we're going to be testing next. So that's enough for our first release. I wanted to take about 10 minutes to present you the 2021 release. I don't know, because we started a little bit late, so I hope this is okay. I'm going to grab the chat to see if there's a complaint on that. Okay, so far, I don't see a moderator telling me that I'm running too late, so I'm going to take this.
Yeah, Dr. Bellec, it's fine. You can go ahead.
Okay, yeah. So I'm going to take another 10 minutes on this 2021 release. So at the current stage, we're able to scan 100 hours.
Sorry to interrupt, Dr. Bellec. Just that there's this black box again in your screen.
Oh yeah, getting rid of the chat, of course.
Right. Thank you so much. Yeah.
So yeah, it's been a challenge from a logistical point of view, but we're now able to scan 100 hours a year. It requires tons of organization because we are currently scanning about each subject gets scanned 3 hours a week. So that's about 20 hours of scanning a week. So we've got a whole sort of like scan crew to make that work smoothly, but we've been doing it now for months. So I'm confident we're going to be delivering 100 hours of data per subject next year. We're going to have a number of like controlled experiments with language, working memory, episodic memory. We're also going to add MEG data and biosignals. But for me, the highlight is that we're currently collecting our first video game data of the Ninja Master, which is a very hard classic platform action game from the 90s on the Sega Genesis platform. So I'm going to show you preliminary behavioral data and our behavioral setup, as well as some preliminary results on like modeling in the video game context. So we use the GymRetro API. So GymRetro is this library developed by OpenAI to train artificial neural network at video games. That library also allows to have humans play and record their game and also annotate the game. So you can annotate in real time what people do and if they lose, else, or their score, or if they die in the game. An engineer, Paul Rimino, has made us some video game consoles that we distribute to the subject. Here you have a picture. It's tiny. You've got the video game controller for scale. But when people started, they can just like select their game and just play like they would on a video game console, except that we record all of the game and we keep track of them. The same soft library can be used to train artificial neural networks, either to play the game and get good at it, or to imitate human behavior. So we really leverage that fantastic work by OpenAI. We had, though, to build our own video game controllers. There's not currently a commercial solution that doesn't involve electronics part, but electronics part disturb the MRI signal and the MEG signal. And it was really not an option to use a controller that would have any metal in it. So André Sears, the engineer of our platform, built a fully fiber optic based video game controller. You see a picture here of it getting assembled. He developed this very interesting design for the buttons. They're independent modules, and you've got one fiber optic bringing light, another one bringing it back to a controller that's electronics based but very distant. And there's this small plastic shutter in between that cut the light and detect button presses. So you can assemble those modules as much as you want, as long as you can deal with the size of the fiber optics. So we've got 8 buttons in our current design. For each button, you have 2 fiber optics, and there's 10 meters between the controller and the controller, the electronic controller and the video game controller. So that's about 160 meters of fiber optics to get that controller to work. It's all 3D printed, and we are going to release all the CAD design for people who would be interested in reproducing this video game controller. We've done about 15 iterations of it, but currently we have a version that works beautifully. Actually, I like it better than the version that we use outside of the scanners. That's a regular, commercial controller. It's very tight. To give you an idea of what Shinobi looks like and what the video gameplay looks like, here's a game. I'm going to actually comment on that game because that's going to be useful a little bit later. So here the subject runs and slash into a ninja, then jumps and does a flying kick into a ninja. That requires a very precise sequence of button presses. That requires a lot of training to manage to do those actions and combine them. Then you've got another run and slash. Once again, that's a pretty tight combination of key presses. And then there's a big hole. And what the player does here is that there's a running, jumping, and double jumping with this like somersault type of behavior. Each of those actions is quite challenging to perform. Combining them even more so, and every failure results in falling into the hole. So it's extremely punishing. So this is a hard game. There's actually still people doing competition of Shinobi 3 into 2020. So it's an interesting, complex task. So here you've got behavioral data of one of our subject learning. Because we piloted on this game, we've been running it for over a year with regular, training. So here you have the probability of a given subject to complete the first level over time with over a year of training, and that's outside of the MRI scanner. So as you can see, the training is like all over the place. It's interesting. Like early, there's like people try strategies and start trying more harder stuff and then fail. So you've got those spikes of completion probability. But eventually they reach this plateau where essentially they complete the level pretty much every time. On the right, you have the completion probability, but in the scanner. So the same subject now is getting scanned, has actually almost finished 10 hours of Shinobi. And as you can see early on, there's a little bit of an adaptation to the new controller. But very quickly, the subjects get back to essentially finishing the level most of the time, like 90% probability of completing that level. That is comparable to what there is outside of the scanner. So that's just one metric we're looking at, but pretty much in every metric, people perform as well inside the scanner with our fiber optics controller as they do outside of the scanner. So one of the first things we've looked at is trying to imitate the behavior of the player with an artificial neural network. So that network works from the video frame. So it's presented with video frame, 20 of them, and then has to decide which button to press. And the network tries to predict what the human's going to do. So it's an approach called human cloning, behavioral cloning, sorry, where you're just trying to reproduce what a human operator has been doing. So the network architecture is fairly standard. It's a convolutional neural net, 5 layers of them, then recurrent neuronal units using a long short-term memory architecture, which is one of the most standard ones, followed by fully connected layer that predict which button combination to press. And here's the artificial neural net playing the game. Here we go. Run and slash. Try to jump and fly kick in the ninja and failed. But the humans fail very often at that stage as well. So not that surprising. It's hard to combine those two things. Run and slash. And now it's trying it. Run and double jump. So it's actually able to, that particular architecture, it's able to learn to reproduce this sort of catalog of human action and really imitate what a particular player was doing. So I would say that our next step is to try to reproduce not only the behavior, but also brain activity. So the way it's going to work, and that's my last slide, is that we're going to be using generative adversarial learning on this task. So there's going to be an artificial neural net that's being fed the video frame and predict button presses, just like what I showed you right now. But it's also going to try to predict brain activity. Likely, we're thinking about using the type of graph convolutional network I've been presenting to you for these additional layers. That's the generator, trying to generate game data and fake brain data. And then there's a discriminator, which is another network. And this one is trying to recognize, it's being presented with example of video game play and brain activity. And it has to decide whether those two data sets jointly come from a human or from the artificial neural net generator. So the discriminator and the generators are trained jointly. And eventually they both become really good, which means that the discriminator can pick little details that give away the artificial neural net, but the artificial neural net become really good at trying to pass for human. So this is our next big step. And one of the key objectives of the project to show we can imitate both brain activity and behavior in video game play. So I'd finally like to thank my institution and all the people involved. You can follow us on social media at CNeuroMod, or check our website, cneuromod.ca, that links to our technical docs, which is already pretty comprehensive. And I'd like to say that this project would have been, I believe, impossible to found using traditional academic funding mechanism, and that the only reason we're able to operate and move really fast on this ambitious project is thanks to a donation for the Kotora Foundation. And you've got Jacques Courtois here in the middle of the picture with our team. So huge thanks to the Kotora Foundation and for your attention.
Thank you, Dr. Bellec. That was really, really interesting talk. I think the inclusion of certain things like friends and certain video games really made it very engaging. I think everybody here liked it very much. So I would like to direct your attention to a few questions that we have had, if that's all right. All right, so I'll just get to it. So the first question was from Ruth. So she is asking that in the in the accuracy of state prediction versus the number of subjects plot, which you showed quite earlier in your talk. So there was this particular mentioning of 7-second window, right? Mm-hmm. So how does the window size matter, or what is the impact of it?
So that's something. So I've linked the paper in the slides, and I'm going to share the slides in the chat after. That's something we've looked carefully into. That was actually one of the primary goals of the paper. You can go down to single frame. so 720 milliseconds in that case, and still get roughly 70% accuracy in the brain decoding. 7 seconds, you start to plateau the quality of your decoding. Now, what you need to bear in mind is that if you follow the time that follows the start of a particular condition, at the beginning, your accuracy is really bad. And you have to wait 3 to 5 seconds before the accuracy gets high. And the reason is simply that in fMRI, we're looking at a vascular phenomenon. It's only indirect reflection of neuronal activity. And that vascular phenomenon takes 3 to 5 seconds to manifest itself. So we cannot really do, instantaneous brain decoding. Maybe we'll be able to do more of that in MEG, but with fMRI, we always have this lag we need to take into account in our models.
Right. Okay. So a latency will always be present, however minor it is. Right.
Yeah.
Thank you so much. Okay, so moving on to the next question. This is from Ayushi Vishnoy. She asks that is the relationship between the signals and brain states one-to-one and onto? Meaning, if you can reconstruct the signals, can you induce a specific brain state?
I've presented 2 projects which are at the moment completely separate. One in which we've tried to look at individual parcellation, get them really reproducible spatially, and look if that improved essentially compression of signal in that reduced space. And our conclusion is that it's possible to have them extremely reproducible and to get much better compression. Now, whether that compression captures information that's useful to do brain decoding, well, we're going to need to plug that into a graph convolutional net and get that answer. And Shima Rastergania is a master student doing that as a project right now, and I still don't have results to present, unfortunately.
Right. Thank you so much. So I think the next question is from Ankush. He asks that how far are we from playing games just using our brains and without a controller?
So I actually, the type of technology I've shown would not be useful to do that. fMRI having this sort of vascular delay, you will need 3 seconds after you're thinking of an action for it to play out, so it just wouldn't work. People would do this kind of there's lots of people working toward that with more EEG. This is not an area I'm particularly familiar with, but I know that for basic games, it already works fairly well. There's actually even like accessories you can buy for I think the Xbox. But I think the degree of control you have is fairly limited. With high quality signal like the one we're going to be acquiring, MEG, that are fairly sort of state-of-the-art resolution. I'm very curious to see how far we can push it, but we haven't collected those signals yet, so I don't have hands-on experience to give you a good answer at the moment.
Right. Do you think personally that nowadays we have machines which operate at 120 frames per second, and we can play games on that, and it's really smooth? So does that help with the latency? I'm not really 100% sure about the concept of it, but do you think that'll help with the choppiness and the lag of the games?
Quite honestly, this is uncharted territory for me. How far in the temporal resolution of the simulation we can get and the brain decoding we can get, I can't tell you. But the truth is that I mean, playing video games, the MEG is not common at all. And I believe the type of hard game we're talking about here it's kind of be the first time it's done. So, I guess we'll figure it out. But I don't know yet.
Right. So yeah, exciting times ahead, I'm sure. Uh, okay, so I think we have another question from Ruth, so which says that if you can shed some light again on parcellation, as to group parcellation versus individual parcellation, and we have another follow-up question, so I'll wait for that. So if you can answer that.
So group versus individual parcellation, and what's the question?
Right, I'll repeat it. So you mentioned about parcellation. So I think she wants you to just shed some light or define what you meant by parcellation,
Oh,
and
okay.
how they differ, the group and individual parcellation. So
Okay. So the parcellation, it can have basically two forms, but the simplest one is simply you've got region and they're being colored. So every part of the brain is in one parcel or not. I use the word parcel because region, people tend to think, like the Brodmann areas. So they're kind of connected. They're a small bit of the cortex, when really functional parcels tend to be distributed and sometimes involve both hemisphere. Actually, most of the time involve both hemisphere. So I just use the word parcel to distinguish from the notion of sort of spatial contiguity. Uh, Parcels can also be probabilistic. So what I was showing was actually probabilistic parcel, where it can be in a parcel with a score from 0 to 1. And they can also be overlapping. And so when you do dynamic parcels of the brain, those parcels end up being overlapping in time. And it happens that those two notions, being probabilistic and being overlapping, are very important to properly capture fMRI fluctuations. And this is, I've been working on that for almost 20 years, and I've always been using, hard parcels, static parcels. That is just one set of parcellation for full-time series. And I recently came to realize that this is extremely limiting and limiting the quality of the compression of the data, as well as the reproducibility of those parcellations. So I think that going for dynamic overlapping parcel is really a key progress to have proper brain graphs at the individual level.
right. Also, what we saw in the slide, that it was dependent on individual tasks. I mean, it was task specific, I believe. So how does it depend on the task complexity, and
Huh.
how will you define task complexity as to what parameters make one task different from the other, or much more complicated than another one? So can you help with that?
Yeah, that's a question I find super interesting. I'm afraid, once again, I don't have a good answer for you, but what I showed you, we generated the parcellation from movie data. And the rationale is that movies are very rich, and you get through all those kind of different situations, different emotions, different levels of engagement. So we figured that there's probably enough in there to characterize the primary modes of functional activity. Our next step, like the type of homogeneity R-square map I was showing, were for other movie data, not the movie data that was used to generate the parcels, but other movie data. But now what we're going to try to see if whether the quality of the R-square is similar for movies of different genres, and also
Exactly.
from different cognitive contexts in the HCP TRT dataset. So in HCP, we have motor and working memory and all that jazz. And I'm really curious to see whether we can also approximate functional activity very well, regardless of that context. But that we are running right now. I haven't seen those results yet.
Right. I think, yeah, it's very exciting if you can, select different kinds of movies. Maybe the timeline of movies, a movie from the 1940s or 50s, they have a very different flavor compared to the movies which are coming out right now. So yeah, that's very
So,
exciting.
so for the final analysis, the plan is to generate the parcellation on Friends, because we have so much of it, and then test how it works on Born Supremacy, Life, Hidden Figures, Wolf of Wall Street, and then all the different tasks of HCP. So we're going to get a good sense about how variable the compression is as a function of cognitive context, and task complexity as well, because some of those conditions, they explicitly vary that. So in working memory, you have 0 back versus 2 back. So you get a direct control on task complexity.
Right. So I think that is all the questions we have right now, Dr. Bellec, and thank you so much again. I just wanted to ask you if somebody else have any other question and they want to reach you. So how to best do that.
So I am on social media. I'm Pierre Bellec on Twitter. Neuromod has its own Twitter account. It's CNeuromod. And if you go on the website, cneuromod.ca, it links to a number of resources, including our GitHub, and you can always post issues on GitHub. So depending on the nature of your question, feel free to contact me, the project, or a specific data set they're listed on GitHub.
Absolutely. I hope everybody enjoyed Dr. Bellec's talk. And Dr. Bellec, again, thank you so much for this amazing, amazing talk.
Thanks for inviting me.
Absolutely.
Thank you for joining in, everybody, and we'll end the session today. We'll start tomorrow again. Hoping to see you all there. Thank you.
Thank you, everyone. Okay, so Dr. Bellec has shared his slides here. So we make sure it is available to everyone. Thank you, Dr. Bellec, again.